This week in longevity research, the age pigment lipofuscin causes oxidative stress, lysosomal dysfunction, and pyroptotic cell death. Authentic cardiac lipofuscin triggers lysosomal dysfunction and pyroptotic cell death in fibroblasts at low concentrations, providing direct mechanistic evidence for how intracellular lipofuscin accumulation causes tissue damage during aging. Brain injury accelerates the onset of a reversible age-related microglial phenotype associated with inflammatory neurodegeneration. Clearing lipofuscin-laden, lysosomal-burdened microglia in old mice restored microglial function and reduced TBI-induced neurodegeneration, directly demonstrating that intracellular waste accumulation in brain immune cells drives age-related neurological decline. Natural killer cell immunotherapy reverses lung fibrosis by eliminating senescent fibroblasts. NK cell immunotherapy targeting the HLA-E-NKG2A axis selectively eliminates senescent fibroblasts and reverses established pulmonary fibrosis, establishing immune checkpoint blockade as a viable senolytic strategy. Fisetin supplementation attenuates premature vascular aging induced by doxorubicin via suppression of cellular senescence and mitochondrial oxidative stress. Oral intermittent fisetin supplementation fully reversed doxorubicin-induced endothelial dysfunction and aortic stiffening, P0.001, by clearing senescent cells and suppressing SASP, providing strong translational support for senolytics as a vascular aging intervention. Fabrication of fusogenic and magnet-responsive cells for transplantation of an intact mitochondrial network. fMRCs enable delivery of whole, functional mitochondrial networks via cell fusion, offering a practical platform for mitochondrial transplantation therapy in diseases driven by mitochondrial dysfunction or age-related mitochondrial decline. Longitudinal autophagy profiling of the mammalian brain reveals sustained mitophagy throughout healthy aging. Widespread age-related mitophagy decline does not occur in the mammalian brain, suggesting that cell and region-specific vulnerabilities, rather than a global autophagy collapse, underlie selective neurodegeneration risk in aging. Secretory form of viral protease, NIA, ameliorates amyloid V pathology and cognitive deficits in a mouse model of Alzheimer's disease. AAV-delivered SecNIA significantly reduced amyloid V plaques and fully restored recognition memory in 5X-FA-D mice, demonstrating proof of concept for enzymatic extracellular AV clearance as a therapeutic approach. Antibodies targeting amyloid V and Tau oligomers in Alzheimer's disease. Bispecific antibodies simultaneously targeting AV and Tau oligomers show enhanced therapeutic efficacy over single-target approaches, representing a meaningful step toward clearing the extracellular and intracellular aggregates central to the amylosens damage category. Genetic extremes, pathogenic progeroid mutations versus longevity-associated protective variants, define a dichotomy that reveals conserved biological pathways, senescence, autophagy, metabolic reprogramming, relevant to aging rate, though without deterministic predictive power for individual trajectories. That is your digest for this week. Stay curious.